run over time. Is that okay with everyone? Okay. So welcome everyone. Today is our second PopBio on the Dark Side seminar of the year 2026, which is the second year that we have this seminar series where we bring together ecologists and evolutionary biologists from this side of the world. And today it is our great pleasure to have Dr. Mayumi Seto to share her research with us. She currently is a professor at the Tokyo University of Agriculture and Technology. I learned that she just moved to this position last year and is starting a new lab right now. Previously, she was a professor at Nara Women's University. And her research combines microbial ecology, community ecology, and biogeochemistry using mathematical modeling. Her recent work focuses on how incorporating things such as thermodynamics during these chemical reactions can shed light onto ecological dynamics, particularly, for example, how considering the chemical networks and the energetic landscapes associated with these chemical reactions can help us understand things such as population dynamics of the microbes that harvest these energies, the community level interactions of the microbial community, making sense of the competition and cooperation that emerge from these chemical networks and also their associated ecosystem functioning and resource utilization and these feedbacks onto the microbial community ecology and evolution. And so I really admire her work because I think it brings a novel perspective showing us that principles in physics and chemistry can indeed shed new light onto ecological dynamics. And her talk today is on a reaction-centric perspective of microbial community networks and ecosystem functions. And so without further ado, please join me in welcoming Dr. Mayumi Seto.
Thank you for your introduction, and please wait a moment. So can you see my slides? Oh, it seems. Oh, can you see my slide?
Yes, we could see.
Okay,
It's great. Yeah.
Thank you very much for giving me the opportunity to speak in PopBio on the Dark Side seminar series. I would also like to acknowledge my funding sources, the Japan Society for the Promotion of Science, JSPS through Kakenhi, and the Japan Science and Technology Agency, JST through the MICASA program. Community ecology aims to understand networks of interactions among species. In this network, species are represented as nodes, and their interactions are represented as links between nodes. These interactions are typically inferred from observations. For example, networks that represent predator-prey relationships are known as food webs. In population dynamics models, each species is affected by others through these links, and as a result, its growth rate changes. In such models, the effects of these interactions on growth rates are summarized and embedded as interspecific interaction coefficients, AIJ here, in generalized Lotuka-Volterra models. In contrast, microbial interactions are often difficult to determine directly from observations because microorganisms cannot be easily observed with the naked eye. Instead, we can focus on which chemical compounds they consume and which compounds they release, and use this information to infer interactions mediated by chemical substances. For example, if species A and species B both utilize glucose, they are expected to compete for that resource, thereby exerting negative effects on each other. In addition, if species A converts glucose into methane and releases it into the environment, it can serve as a substrate for species C that utilizes methane, thereby positively affecting the growth rate of species C. In this way, by defining links between microorganisms and chemical compounds, and then reducing chemically mediated links into direct links between microbial nodes, we can construct a microbial interaction network analogous to those well studied in macroecological systems. This enables us to apply theoretical frameworks developed for macroecological networks to microbial communities. However, this reduction process inevitably leads to a loss of information we can gain from chemistry's perspective. Chemical compounds are not merely passive mediators. They contain essential information that directly constrains microbial growth. Organisms take up substances required for growth and excrete those that are unnecessary, and this selection is determined by their metabolic capabilities. For example, humans take in organic carbon and oxygen, synthesizing ATP, and release carbon dioxide and water. Because our energy metabolism depends on these substrates and our metabolic pathways. Similarly, sulfate-reducing bacteria, this blue guy, rely on metabolic pathway that use sulfate ion SO4 2- to generate ATP and therefore take up sulfate from their environment. The type and amount of substances utilized ultimately determine the amount of energy gained, which directly controls growth rate of microorganisms. It is similar to our daily life decision on what we eat. We estimate energy intake based on calories levels on food and use this information to anticipate changes in body weight. When managing body weight, we often check the calorie content per unit mass on food packing. For instance, one might think, wow, a Big Mac meal contains around 1,000 kilocalories. Perhaps I should skip the set and just order the burger. In other words, we implicitly predict the rate of weight gain from the type and quantity of substances we consume. Similarly, in microbial systems, information on which metabolic reactions are utilized enables prediction of growth rates through energy yield. Importantly, this reaction-centric perspective influences not only the growth of a single species, but also microbial interspecies interactions and community structure. For today's talk, I propose a framework that explicitly links microbial networks to metabolic reactions and interprets microbial interactions and community structure based on a reaction-level linkage structure, namely a reaction-centric approach. In this presentation, I will first introduce the basic concept of this framework. Next, I will explain how the reaction-centric approach enables the evaluation of growth rates and the prediction of microbial niches. Finally, I will discuss emerging questions revealed by this framework, including the diversity of metabolic evolution and the relationship between biodiversity and ecosystem functioning. First, I will introduce the framework of the reaction-centric approach. A reaction-centric approach explains microbial interactions as emergent from metabolic reactions. In conventional community ecology, we typically start by listing organisms. Here, B1, B2, B3. Please consider these are the name of bacteria and their relationships, and then construct a microbial interaction network like this. These are the interaction of these B1 to B3. In contrast, in the reaction-centric approach, we first describe the set of metabolic reactions in the system. Reactions 1, 2, 3 in the figure on the right represent simplified metabolic reactions, and the network below illustrates their structure. Since each microorganism can be linked to specific reactions based on its metabolic traits, we can derive a microbial interaction network. The key difference is that interactions are not assumed, but emerge from reaction networks. Here, I outline the basic workflow of the reaction-centric approach. In the first step, we select the set of metabolic reactions of interest. These may include reactions that can occur in the environment or those that microorganisms are capable of utilizing. It is also possible to focus on a subset of reactions depending on your research question. In this sense, this step essentially defines the target community of interest. In the next step, we establish the mapping between reactions and microorganisms. In other words, we identify which microorganisms are capable of utilizing which reactions. In the third step, we describe interactions among microorganisms based on these reactions. For example, competition arises when multiple species utilize the same substrate, whereas positive interactions occur when the product of one reaction serves as a substrate for another microorganism. These interactions can be evaluated based on basic principles in community ecology. Finally, in step 4, we use these reaction properties to understand community structure and its characteristics. There are many types of metabolic reactions, and the choice of which processes to focus on depends on which community property we are interested in. For example, if we are interested in antagonistic traits, we may focus on reactions involved in the production of toxins as well as the corresponding defense metabolism. In the study shown on the left, microbial interactions are mediated through a sequence of metabolic reactions, including the production of toxin and its detoxification. In my case, I am interested in how communities use energy, so I mainly focus on energy metabolism to construct the network. The figure on the right shows some examples of species interactions when we focus on energy metabolism. In the next slide, I will briefly explain the energy metabolisms of microbes in more detail. In human metabolism, as you know, energy is obtained through aerobic respiration. In this process, organic compounds, which correspond to food, act as electron donors, and oxygen act as the final electron acceptor. Electrons derived from organic compounds are transferred through the electron transport chain and eventually accepted by oxygen. The amount of ATP produced depends on how many electrons flow through this system electron transport chain. However, in contrast to animals, which rely on a fixed combination of organic matter and oxygen, bacteria and archaea exhibit a much greater diversity in their energy metabolism, the choices of electron donors and acceptors. They can use a wide range of electron donors and acceptors, and different combination of these allow electrons to flow through the electron transport chain, like our electron transport chain. The table on the right shows some example of such combinations, although there are more energy source reactions utilized by microbes. As a result, even when we focus on energy metabolism only, the resulting reaction networks and therefore the community structures derived from them can be highly diverse and complex. Returning to the workflow of the reaction-centric approach, the next step is establish the mapping between reactions and microorganisms Here, I use sulfate reduction as an example. This reaction is part of energy metabolism. Its final product, sulfide, HS-, can become hydrogen sulfide, which is toxic to surrounding organisms. In this process, sulfate SO42- is taken up by the cell and is converted step by step into sulfide. Each step is catalyzed by specific enzymes like SAT, APR, and DSR. Therefore, a microorganism can perform this reaction only if it possesses all the genes required to express these enzymes. Based on this idea, we can map a reaction to microorganisms that encode the complete set of required enzymes to a reaction. Inferring microbial functions from the presence or absence of functional genes is already a well-established approach in metagenomic analysis. The figure on the left shows an example where genomes are reconstructed from environmental samples called MAG were examined for genes involved in sulfate reduction. The genes highlighted by red box here and here represent those required for this function. In other words, microorganisms that possess these genes can be associated with a sulfate reduction reaction. The reaction on the right is a less strict representation, but it shows a network linking lineages to reactions. The numbered groups indicate associations between reactions and microorganisms. At a conference about 2 weeks ago I attended, I was told that these groupings may correspond to ecological guilds. Indeed, this mapping is closely related to the concept of ecological guilds in the sense that microorganisms are grouped by their roles. However, since guilds are typically defined based on trophic positions, it may be more accurate to describe this grouping as a functional grouping based on shared reactions. So far, we have established the mapping between reactions and microorganisms. From here, we move on to understanding interactions among species. This slide shows a process called nitrification. Nitrification is one form of microbial energy metabolism, in which ammonia, NH3, is converted to nitrate, NO2-, and then to nitrate, NO3-. In aquatic systems, an increase in nitrate often leads to nutrient over-enrichment and eutrophication, so controlling the level of nitrate is important for aquatic ecosystem management. While some microorganisms can perform all of these reactions within a single cell, the upstream reaction and the following reaction, in many ecosystems, especially in terrestrial ecosystems, the upstream and downstream reactions are carried out by different groups of microorganisms. Let us now consider the interactions between these two groups. Microorganisms performing the upstream reaction produce nitrite, NO2-, which serves as a substrate for downstream microorganisms. Therefore, they have a positive effect on the growth of the downstream group. At first glance, the downstream microorganisms may seem to have no effect on the upstream group, since they do not supply any substrate. However, in terms of energy acquisition, the downstream microorganisms can actually have a positive effect on the growth of the upstream group. This idea comes from my research collaborator, Yo Iwasa, and highlights how reaction energetics shapes growth. From here, to explain this mechanism, I will move on to how we can predict growth and ecological niches based on reaction energetics. To discuss reaction energetics, we first need the concept of Gibbs energy. I will not go into a strict definition here, but you can think of Gibbs energy as a measure of how much energy can be extracted from a system under constant temperature and pressure. Whenever a chemical reaction proceeds, it is accompanied by a change in Gibbs energy. We usually denote this change delta Rg or delta Gr. In general, systems evolve in the direction that decreases Gibbs energy. For example, photosynthesis from right to the left does not proceed without the presence of light, even if carbon dioxide and water are present. This is because the reaction requires an external Gibbs energy input, meaning that delta Rg, the sign of delta Rg is positive. In contrast, aerobic respiration from left to the right proceeds spontaneously when organic matter and oxygen are available. In this case, Gibbs energy decreases through the reaction, and the sign of delta Rg of this reaction is negative. Living organisms can convert part of the released Gibbs energy into ATP. Therefore, the magnitude of negative delta Rg provides an upper bound on the energy that microorganisms can obtain from a reaction. So far, I have explained that delta Rg serves as an indicator of energy. Now, I will briefly explain how we actually calculate it. I will skip the detailed derivation, but if you are interested in the calculation, please feel free to ask me later. Here, let us consider a simple reaction where 2 moles of A converted into 1 mole of B. These are molecules. In this case, the Gibbs energy change of the reaction, delta Rg, is given by the difference between the chemical potential of the product B and that of the reactant A. The chemical potential can be thought of as the Gibbs energy per unit amount of substance. So delta Rg represents the energy difference associated with producing 1 mole of B and consuming 2 of A. By rewriting this expression, we obtain the equation shown below. The first term, delta Rg circle, is a reaction-specific property or the substance-specific property and can be obtained from thermodynamic databases. The second term captures the effects of environmental conditions, such as concentration and temperature. This means that the energetic favorability of the same reactions can change depending on the environment. A key point is that by combining database values with environmental conditions, we can quantitatively evaluate delta Rg under given conditions. Here, I briefly introduce some commonly used tools for calculating delta Rg. Shinosu's CHNOS is an R package that allows thermodynamic calculations mainly for aqueous species and many inorganic reactions. It is very convenient, although its coverage of solid phases is somewhat limited. The other Geochemist's Workbench, or GWB, is a commercial software package. It provides comprehensive thermodynamic databases with particularly strong coverage of minerals and solid phases. Equilibrator is a tool designed for biochemical compounds. It estimates thermodynamic properties using root contribution methods for chemicals and can be assessed through both web interfaces and Python. So each of these tools has its own strengths, and it is important to choose them depending on the target systems or reactions. We are also developing a pipeline tool to automatically evaluate delta RG for multiple reactions under given environmental conditions. We start from a biogeochemical reaction database. In this case, we compiled more than 2,700 reactions, including reactions from previous studies. Then, using our modules, we first organize and generalize the reaction set in a consistent format. At the same time, we use observed environmental data such as concentrations, temperature, pressure, and ionic strength. Then, using the thermodynamic databases, we obtain thermodynamic properties such as standard use energies and activity coefficients. Then finally, these inputs are combined in our calculation module. Then we can get the reaction energetics change over time or across environments, as shown in this example. We are currently preparing this for submission next month, but if anyone is interested in using it at this stage, please feel free to let me know. I would like to use your data as the input data or test data for this pipeline tool. So far, I have explained how we compute delta RG in practice. Now I would like to show how we can use this information to predict microbial growth. A fundamental model of a microbial growth, the most well-known one, is the mono model, where a growth rate depends on a substrate concentration. Since its proposal, many studies have attempted to decompose and reinterpret the parameters of the mono model. In particular, some extensions focus on the energy obtained from substrate utilization, where delta RG is explicitly linked to growth rate, and this is one example from our previous study. Here, the growth rate is decomposed into two components: kinetic constraints and thermodynamic constraints. Kinetic constraints describe how fast the reaction can proceed. In other words, how much substrate can be processed per unit time. To give an intuitive example, kinetic constraints are like how much food you can eat per day. In contrast, thermodynamic constraints describe how much energy can be obtained from those reactions, like how much energy you get from that food. Even if you can eat a lot, if the food contains little energy, your growth will still be limited. As I mentioned earlier, this energy depends on the combination of electron donors and acceptors, as well as the environmental conditions such as concentrations. In environments where these factors strongly limit growth, thermodynamic constraints become essential. One interesting aspect of delta RG is that the energy obtained from a reaction depends not only on the amount of substrate, but also on the amount of reaction byproducts. The left figure illustrates this relationship. The x-axis shows the ratio of B to A, and the y-axis shows minus delta RG of the reaction from A to B. When the concentration of B becomes relatively high compared to A, the energy that can be obtained from the reaction decreases. The yellow region represents a critical energy threshold required for ATP synthesis noted by previous study. So depending on the reaction, the accumulation of B or byproduct can therefore become limiting or even detrimental for survival. This provides the mechanism behind what I mentioned earlier. Microorganisms performing downstream reactions can positively affect the growth of upstream microorganisms. The right panel shows simulation results for two functional groups. When functional group 1 is cultured alone, it cannot grow under these given conditions and eventually declines. However, in co-culture with functional group 2 consumes B, which increases the energy yield of group 1. As a result, group 1 can survive with group 2. This suggests that interactions mediated by reactions are governed not only by material exchange, but also by energy transfer. Now, an important point is that the impact of the reactant product ratio on growth depends on the type of energy yielding reaction. We can think of delta RG as consisting of two parts. One term, the first term, does not depend on concentrations, and the second term does depends on concentration. The second term varies with temperature, but under typical environmental conditions, its magnitude is on the order of tens of kilojoules. So if the first term is much larger than that, the effect of a concentration change is relatively small and does not strongly constrain growth. For example, in aerobic respiration, which is our primary energy source, the first term is about 2,800 kilojoules. So thermodynamic constraints are relatively weak. In contrast, for reactions lower in this table, the first term becomes much smaller and the second term becomes relatively important. This means that for low energy reactions, the reactant product ratio can strongly affect growth. Here, reactions are ordered by the magnitude of the first term. If all concentrations were equal, reactions at the top would provide higher energy, while those at the bottom would provide less. So if we think of reactions as ecological niches, the negative of delta RG can be interpreted as a measure of the quality of that niche. And this suggests that it can be used to predict microbial presence and functional expression. Based on this idea, a number of studies have attempted to predict microbial niches by calculating delta RG from observed environmental conditions. I have also worked on this topic, evaluating whether microbial growth is energetically feasible on Mars under energy-limited conditions. This was a collaborative study with Philip Van Katlin at the University of Waterloo and Katsuhiro Noguchi at Nara Women's University. However, in complex nature environments and with diverse microbial communities, the extent to which delta RG can actually predict microbial presence and functional expression is still not well established. Most of the existing evidence is indirect. So there is a strong need for quantitative validation based on observational data to assess the predictive power of delta RG. Given that I have been working on theoretical calculations of delta RG, I felt it was important to test by myself whether these predictions actually hold in real systems. To do this, I designed a mesocosm experiment where we simultaneously measured the chemical species and microbial community data to evaluate whether delta RG can predict microbial presence and functional expression. This experiment was carried out by Mana Ito and Katsutoshi Ito at the Japan Fishery Research and Education Agency. They conducted an extremely demanding sampling campaign, sampling every 3 hours continuously for 11 days. So I'm quite I quite admire their enthusiasm to this work. Thank you, Mana-san and Katsutoshi-san. We also collaborated with many researchers for chemical and microbial analysis, which allowed us to systematically examine the relationship between the ecology and microbial communities. The results are not yet published, so unfortunately, I cannot show them here today. But if you are interested in, I would be very happy to share updates in near future. Finally, I'd like to present two insights into questions that could only be addressed through a reaction-centric approach. One on the evolution of energy metabolism, and the other on the relationship between energy metabolic diversity and community-level energy use. First, I will talk about a question related to the evolution of energy metabolism. The figure on the left shows the nitrogen cycle observed on Earth, while the figure on the right represents all theoretically possible nitrogen transformation reactions as the combination of the functions of enzymes as a network. Most of these transformations are driven by microorganisms, and many of them are associated with energy metabolism. At first glance, nitrogen metabolism appears highly diverse. However, from a chemical perspective, many more reactions are actually possible, and when we include all of them, we obtain a highly complex network links like this one. But when we compare this theoretical chemical network with the reactions that microorganisms actually use, it appears that only a subset of reactions is selectively utilized. This could simply be due to historical contingency, but it also raises the possibility that evolutionary selection favors reactions that are energetically more advantageous at the community level. To test this idea, we first enumerated all chemically possible nitrogen reactions, around 1,000 reactions, and evaluated their energetic favorability under environmental conditions. We then compared these with observed microbial nitrogen metabolism. We found a strong correspondence. Energetically favorable reactions are preferentially utilized by microbes. This suggests that the energetic structure of reaction space may constrain the diversity of nitrogen metabolism. This work was done in collaboration with Ryuhei Nakamura and Hideshi Oka at Riken and Risa Sasaki from my research lab. Finally, I'd like to introduce our theoretical work with Michio Kondo at Tohoku University on the relationship between community structure and energy use efficiency in metabolic networks. We developed a community dynamics model based on the reaction-centric approach, which we call the eco-redox model, where reactions and microbial functional groups are explicitly linked. In this model, adding reactions automatically introduces corresponding functional groups and expands the system dynamics like this. Our simulations show that a higher metabolic diversity, as shown on the x-axis, leads to increased biomass production efficiency and energy use efficiency, as shown by bright colors at the community level. Importantly, this improvement of biomass production and energy use efficiency is associated with the emergence of cyclic structures in the reaction network. This suggests that internal recycling within the community enhances material reuse and allows more efficient energy utilization. This is the final slide of my talk. To summarize, first, by applying reaction-centric approach, microbial interactions emerge from metabolic reaction networks. Second, The change in Gibbs energy denoted by delta RG helps us predict growth and ecological niches. And finally, the energy structure of reaction space shapes biodiversity and ecosystem function. In other words, microbial ecosystems are strongly shaped by the energetics of reactions. Thank you for your attention. I would be happy to take any questions.
Right. Thank you, Mayumi, for the great talk.